Hello everyone, welcome back to the session. So in the yesterday's class we have completed the cell division. So up to the cell division we have completed all the most important topics regarding the cell biology. So in today's class we are going to start a new chapter, biochemistry, and in the biochemistry we have to discuss about carbohydrates, um, proteins, lipids, vitamins, minerals, and all those things. And the first most important topic will be the carbohydrates. So we are going to discuss about that. So what is carbohydrates generally? So carbohydrates are nothing but a sugars. So when we chemically defined this particular carbohydrates, this carbohydrates are polyhydroxyaldehydes or polyhydroxyketones or substances that yield them on hydrolysis. So which substance, the substance which is undergoing a hydrolysis process, and they are giving the sugar. So those substances we are calling them as carbohydrates. But generally you should know that carbohydrates are nothing but a sugars, and we can say this carbohydrates as polyhydroxyaldehydes and polyhydroxyketones, right? This is very important. And what will be the general molecular formula for the carbohydrates? The general formula for carbohydrates will be CH2O of N. So this will be the basic formula of carbohydrates. So we are having a monosaccharide, disaccharide, polysaccharide, all those classification under the carbohydrates. But the general molecular formula for carbohydrates is CH2O of N. So this will be the molecular formula of monosaccharides. And you should know, see here, so in the general formula, so in the formula we can see what are all the elements: carbon, hydrogen and oxygen. So carbohydrates, in the sense you should know there is a presence of carbon, hydrogen and oxygen, isn't it? So this carbon and hydrogen and oxygen with all its variations and mixtures, we are saying that as a carbohydrates. Just see, look at the picture, and what will be the ratio of hydrogen to oxygen? If it is in a monosaccharide, the basic general formula will be 2 hydrogen and 1 oxygen. So that is what the hydrogen is to oxygen ratio will be 2:1. So all these are very important basic information about carbohydrates. So how we can expect the question from the basic information? Not all carbohydrates strictly follow CH2O of N. Say for an example, deoxy sugars. So we are having deoxy sugars. Deoxy in the sense absence of oxygen in the sugar. So that is what we are saying, deoxy sugars. So those sugars which we are saying that as a deoxy sugars, those sugars are not obeying this general formula. Why? Because in this general formula we are having carbon, hydrogen and oxygen, and in the deoxy sugars, in the sense absence of oxygen. So that is why the deoxy sugar should not follow the general formula. So that is why the statement, not all carbohydrates strictly follow the general formula of CH2O of N. So this is the very important slide, kindly make a note of it. Now, so these are all the different types of carbohydrates. We are going to discuss about that, but before that you should know what will be the linear structure of all these carbohydrates. Just look at this: allose, maltose, glucose, mannose, glucose, idose, galactose, talose, and psychose, fructose, sarbose, tagatose. So all these were the carbohydrates. Basically we know only glucose, galactose, fructose and all those things, but you should know all these were the different types of sugars which is coming under the major classification called carbohydrates. And before that we should know the general formula for glucose. So this is how glucose will be. What will be the general formula for glucose? C6H12O6, isn't it? C6H12O6. Now look at this. This point is one carbon, and here one carbon, here one carbon, here one carbon, and here one carbon. So all these were the place of a carbon, and we are having a hydrogen and we are having a glucose. See, just look at this. Why we are saying this as an alpha? What are all the sugars we are saying beta, and what is D glucose and what is L glucose? We are going to see one by one. As of now, just know all these these are all the basic structures. Just know the basic information about the carbohydrates. Now, how can we classify the carbohydrates? Based on the hydrolysis process, we can classify this carbohydrates into monosaccharides, disaccharides and the polysaccharides. So this is based on the hydrolysis. So see here, monosaccharides. So based on the hydrolysis, carbohydrates can be divided into three broadly: monosaccharides, disaccharides and polysaccharides. Now what is monosaccharides? Monosaccharides cannot be hydrolyzed further. This will be the simplest form of carbohydrates. The simplest form of carbohydrates will be the monosaccharides, which means we can consider this type of sugars as monomers. What will be the texture of this monosaccharides? Sweet, crystalline and water soluble. These monosaccharides will be very sweet, and the shape will be crystalline in nature, and whether it is soluble or insoluble, it is water soluble. And what are all the examples of this monosaccharides? That will be glucose, fructose and ribose. All these were the examples of this monosaccharide. So this is what a monosaccharide. So see, just look at this picture, glucose and fructose. So this is what the structure of glucose. Just look at this formula, C6H12O6 and C6H12O6. So both of these glucose and fructose are considered as a monosaccharides, and these two monosaccharides together we are calling them as a disaccharide, which is nothing but a sucrose, isn't it? So this is what a glucose, a fructose and sucrose. See, there is a 5 carbon ring, 1 2 3 4 5 and 6. C6H 1 2 3 4 5 6 7. So some of this hydrogen has been it is not visualized in this picture, so that is in the cyclic ring, it is not there. See, C6H12O6, C6H12O6. So together we can add by adding both of these two, we can get C12H22O5. So we can call this particular sugar as a sucrose. So both glucose and fructose is linked by a linkage, and one hydrogen has been removed as by binding of these two sugars, a water molecule has been eliminated, and this particular process is called as a hydrolysis process. So based on this hydrolysis, the carbohydrates has been classified into mono, di and polysaccharides, right? Now, oligosaccharides. So, monosaccharides cannot be hydrolyzed further, but in disaccharides, see, oligosaccharides means disaccharides. So, disaccharides are most important. Why? Because, see, when we hydrolyze the sucrose, on hydrolyzation of the sucrose, we can get glucose plus fructose. On hydrolysis, if a particular sugar gives rise to two monomers, then it is called as a disaccharides. So say for an example, maltose. If we hydrolyze maltose, we can get glucose plus galactose. Glucose plus glucose. If we hydrolyze lactose, we can get glucose plus galactose. So when we hydrolyzing all these sugar, on the process of hydrolysis, they yield two monosaccharides. So that is why we are saying this particular sugar as a disaccharides. Now, polysaccharides. 10 on hydrolyzation, this particular sugar can give rise to 10 monosaccharides. So that is why we are keeping this particular sugar under the polysaccharides. So usually what we have discussed with respect to the monosaccharides, those sugars are very sweet, but here it is tasteless, and monosaccharides were soluble in water and polysaccharides are insoluble in water. Now what are all the examples for this polysaccharides? That will be starch, sugar, cellulose and chitin. So all these were the best example for this polysaccharide. So in an exam point of view, they usually ask these type of questions in a mass of following questions. Match examples with reducing and non-reducing nature. We are going to discuss what is reducing and what is non-reducing. So based on the functional group, the carbohydrate can be classified into two. One is aldoses and the ketoses. Just listen to me. So first we have studied what will be the general features of this carbohydrates, and whenever it comes to the classification of the carbohydrates, based on the hydrolysis we have classified the carbohydrate into monosaccharide, and we have discussed about the monosaccharide with examples, and disaccharides with examples we have discussed, and polysaccharides. So what is polysaccharide, what will be the basic features of it, and what will be the examples of this polysaccharide. So these were all the types of the carbohydrates. Now, based on the functional group, this carbohydrates can be classified into two groups. One is aldoses and another one is ketoses. So the aldoses, they contains aldehyde group. So in aldoses they contains an aldehyde group. The examples of aldoses will be glucose and ribose. So the ketoses, they contains ketone group, and the example will be fructose and ribulose. So if any sugar they contains an aldehyde group, they are coming under the category aldose sugar, and example will be glucose and ribose. See, if any sugars have this keto group, then we are calling this particular sugar as ketose sugar, and the example will be fructose and ribulose. So see here, identify aldose versus ketose using carbonyl position. So the question can be asked from here. We are going to discuss all those things. See, so based on the carbon atoms, we can classify the carbohydrates. If they are having 3 carbon, we are calling them as a triose, and the example for the triose sugar will be the glyceraldehyde. We are going to see the picture. And tetrose, if they are having a 4 carbon, we are calling them as a tetrose, and the example for the tetrose will be erythrose. And if the carbon, if there is a 5 carbon, then the 5 carbon sugar will be coming under the pentose sugar, and the best example will be the ribose. If the carbon number is 6, then we are calling that particular sugar as a hexose sugar. The example will be glucose and fructose. If the carbon number will be 7, we are calling them as a heptose, and the example will be the pseudoheptulose. Now, pentose is in the nucleic acid. So this can be expected in the examination. Why? Because we are having two types of nucleic acids. One will be DNA and RNA. And both of these have, so DNA, in the sense, the DNA is, how can we define the DNA? It is a polynucleotides. So in a polynucleotide, so many nucleotides together we are calling them as a polynucleotide, and a group of polynucleotide we are calling them as a deoxyribonucleic acid. See, what is nucleotide? Base, sugar and phosphate. And here comes the sugar, and this is nothing but a carbohydrate. The sugar which is present in the nucleotide of the nucleic acid is the ribose sugar. And the ribose is coming under 5 carbon category, and we are calling them as a pentose. So that is why pentose is in the nucleic acid. So this can be expected in the examination. Now these are all the structural formula for the triose, tetrose, pentose, hexose and heptose. Just look at this picture. 1, 2, 3. Okay, so 1 carbon, 2 carbon and 3rd carbon. So this, as there is a 3 carbon containing sugar, we are calling this particular structure as a triose. Just look at this picture. So the simplest molecular formula of this is, just look at this, CHO, CHOH. So 1, 2, 3. So this is very tricky to just listen. Just listen this structure. CHO, aldehyde group. So we can classify this sugar under the aldose sugar. And we are having 3 carbons, so we can say that this particular sugar as triose. Just look at this picture. CHO, CHOH and CHOH, and here CH2OH. So as there is a 4 carbon, we are saying this particular sugar as a tetrose, and we are having an aldehyde group, so it is an aldose sugar. Again here it will be the CHO and CHOH, 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 and the last will be CH2OH. So as it have 5 carbon containing compound, it is a pentose, and they are having an aldehyde group, so it is coming under an aldose sugar. Now just look at this picture. 1, 2, 3, 4, 5, 6. So 6 carbon. So we can classify them under the hexose. Now again see here 1, 2, 3, 4, 5, 6. Again this carbon is also having the 6 carbon, 6 carbon. But look at this picture. This is very important here. So just look at this. See here. What will be the functional group present in this sugar and what will be the functional group present in this sugar? So both are having the 6 carbon containing compound. So both will be coming under the hexose category. Now the functional group present here will be the ketone and the functional group present here will be the aldehyde. So we can call this particular sugar as an aldose sugar and this sugar as a ketose sugar. Clear? Now, so you should know what now we have discussed what will be the aldose sugar and the ketose sugar based on the functional group and based on the carbon atoms, based on the number of carbon present in the sugar, we are classifying them as a triose, tetrose, pentose, hexose, heptose and all those. Now, so we can based on the configuration, we can classify whether this particular sugar is a D or L configuration. So we have studied in the previous slide alpha D glucose and beta D glucose. How we can say that this particular glucose in a D configuration and L configuration? We are going to discuss about that. Just look at the picture here. Based on the position of OH on the penultimatum carbon. So you should know that based on the position of OH group is deciding whether this particular sugar is in the D configuration or L configuration. So if the OH group is in the right side, then we can call them as a D configuration. If the OH group is in the left side, we can say that particular sugar as an L configuration. So just look at this picture. Biological sugars are mostly D isomer, not L isomer. This can be expected in the exam. And D is not equal to dextrorotatory. Optical activity here will be different. Let me explain you a little more elaborate. Just look at this picture. D and L configuration of the carbohydrates. So, but before that, you should know what is meant by chiral center. So, this is a carbon. And here it is an oxygen and here it will be a hydrogen and say here it will be okay. Just look at this picture. If any carbon containing all the 4 sides are different, then it is called as a chiral center. Okay, so just listen here. Locate the chiral center farthest from the carbon, that is the ketone group. So just look at this picture. So here is different and here it is different, here it is different and here it also will be different. So if the carbon is surrounded by a different sites, if a carbon is associated with the different groups, then that particular carbon is called as a chiral center. In that chiral center, in that chiral carbon, if the OH group is in the right side, then we are calling that particular sugar as a D sugar. And in that particular chiral carbon, if the OH group in the left side, then we are calling them as the L sugar. So this is very, very important. See, let me explain you once again. So what you should know what just look at this. Just look at this. Okay, so here also CHOH, here also CHOH, here also CH2OH and here CH2OH. There will be no chiral carbon in this. So if a carbon is associated with if the carbon is associated different groups on all the sides, then we are calling that particular carbon as a chiral carbon. And in that chiral carbon, if the OH group is present in the right side, then we are calling them as a D sugar. If in the chiral carbon, if the OH group is in the left side, then we are calling them as a L sugar. Just look at this picture. See, presence of a chiral carbon. So to identify the chiral carbon, so it is very difficult to identify a chiral carbon. So until we are in a clear idea about the chiral carbon, how to find it. So the number of optical isomers is equal to 2 power n. So to identify the chiral carbon, we have one formula. So that is 2 power n, which means, so you should know, so to identify the number of isomers. See, why I am saying like this is, so you have to know what is meant by isomer. So isomer means they are, see, it is D glucose and L glucose. Their OH position will be different. The OH group will be in the left side and it will be in the right side. So this is what so based on this, we can identify whether it will be the D glucose or L glucose. But you should know that whether it will be the, see here, the same molecular formula, but different structural, same molecular formula but different structural formula, that is called as an isomer. So to identify the isomer, you should know there will be one formula. To identify the isomer, we are having a formula. So that formula will be 2 power n. So in the place of n, you should know, you have to calculate what will be the number of chiral carbons present in that particular structure. Say for an example, in a glucose, there will be a 4 chiral carbon, and if there will be a 4 chiral carbon in the glucose, then what will be the number of isomer of this particular glucose? So to identify the number of isomers, you should know how many number of chiral carbon will be present in the structure of the glucose. So as they have given here, we are having 4 chiral carbons in glucose. So as it is a 4 chiral carbon, the 2 power 4, then 2 power 4 is equal to 16. So the number of isomer present in glucose will be the 16 isomers. So this is very, very important in an exam point of view. For that, you should know how many chiral carbon will be present here. So locate the chiral center farthest from to identify this D and L glucose. Okay, so all these were very important. Just make a note of it. Now, epimers. See, epimers differ at one chiral carbon except anomeric carbon. So we are having an isomer. That is, this is the isomer. Same molecular formula but different structural formula. Same molecular formula, C6H12O6 for both D glucose and L glucose. C6H12O6 for D glucose as well as the L glucose. But the structure has been different. Just look at this here. So all the OH group, just look at this H. Here OH will be in the right side and the H will be in the left side. So the structural formula has been different, but the same molecular formula but different structural formula. So this is what an isomer. So to identify the number of isomers present exhibited by this compound, you should know what will be the number of chiral carbon. If there will be the number of chiral carbon is known by you, then you have used the formula 2 power n. In the place of n, you should know, you should write what will be the number of chiral carbon. So if there will be the 4 chiral carbon present in the glucose, then the number of isomer present in glucose will be 16 isomers. Now comes to the epimer. So differ at one chiral carbon except the anomeric carbon. So just look at this picture. The glucose and mannose, just see here. Okay, just see here, this is an epimer. Only one chiral center with different configuration. Just look at this D glucose and just look at this D mannose. So glucose and the mannose. So just here an aldehyde group and here an aldehyde group. And here CH2OH, here also CH2OH. And here HOH. Okay, here also HOH. And just look at this, H here, H is on the left side, OH is on the right side, and here OH is on the left side and H is on the right side. And only in the second carbon, only at the second carbon, that is only only one chiral center with different configuration, all other carbons will be present in an equal configuration. So that is why we are calling them as an epimer. So all these were a chiral carbon. See, in this chiral carbon, we are having carbon, OH and CH2OH. And see, in this carbon, we are having OH and H and OH. And here OH and H. And here H and OH. So all these were considered as a chiral carbon. So in this chiral carbon, and only one chiral carbon, we are having the different configuration. All other chiral carbon is associated with the same configuration. So that is why we are saying that as an epimers. Right? So this is what the epimers. So the differs at only one chiral carbon except the anomeric carbon. Just look at this, look at the example. So this is very important. This is what an exam favorite. So identify the epimer pair. So if in the glucose and mannose, the second carbon will be different. So that is why we are saying C2 epimers. In the glucose and galactose, in the fourth carbon, the configuration has been different. So that is why we are saying this as a C4 epimer. Now coming to the anomers. So here differ at anomeric carbon, C1 in aldose, alpha glucose versus beta glucose. Just look at here, there is an alpha glucose and beta glucose, and the anomeric carbon, at the anomeric carbon in the sense C1 carbon, at the first carbon position, if the OH is in the downside, then we are calling them as the beta glucose. If the OH group is on the upper side, then we are calling them as a beta glucose. If the OH is on the downside, then we are calling them as an alpha glucose. And if the OH group is on the downside, then we are calling them as a beta glucose. Just look at this picture. In the first carbon, the OH group in the downside. And here also in the first carbon, we are having the OH in the downside. So we are calling them as an alpha anomers. Now, in the beta anomers, the OH is on the upper side. Then we are calling them as the beta glucose. So, this is very important. Now, the ring structure of the monosaccharide. So, the ring structure in the sense, so this is what a ring structure. The ring structure of the monosaccharide. So, cyclization, aldehyde or ketone reacts with the OH, forms hemiacetal, that is aldose, or hemiketal or ketose. So the aldehyde group or ketose group, if it is react with the OH, the linear structure, then it will be, the linear structure will be turned to a cyclic structure. So that is why we are saying that as a hemiacetal or hemiketal. So the ring times will be pyranose and furanose. So this is very important. See here, if there is a 6-membered ring, 6 carbon membered ring, then we are calling them as a pyranose. Example, glucose. If there is a 6-membered ring, then we are calling them as a furanose. Example, fructose. Haworth or Fisher projections. Fisher projections means this is what a Fisher projections. Now this carbon, it will be associated with this OH. Then as a fusion of these two, it will be shown as a cyclic structure. Just look at this picture. So this is what a cyclic structure of glucose, and this is what a cyclic structure of the fructose. Just look at this picture. This is a OH and H group, and here the H group and CH2OH group, and this is how the structure of glucose and fructose. And the basic difference between this glucose and fructose will be here 1, 2, 3, 4, 5, 6. 6-membered ring. So this glucose is considered as a pyranose ring. 6-membered ring. Now in the fructose, they are having a 5-membered ring, that is 1, 2, 3, 4, 5. 5-membered ring. So we can call them as a furanose ring. So example will be the fructose. So this is very important. Now coming to the reducing and non-reducing sugars. So in case of reducing sugars, there will be a free aldehyde or ketone group. So free aldehyde or ketone group. Just look at this picture. So let me explain you after this. see here, free glucose and free ketone group. So if the aldehyde group, if the functional group, that is the aldehyde or the ketone, if it is in the free end, then we are calling them as a reducing sugar. And these reducing sugars, whenever they are undergoing this Benedict test and failing test, they will be give the positive results. So this is what those sugars which are giving a positive result for this Benedict test and the failing test, we are calling that sugar as a reducing sugar. And the example for this reducing sugar will be glucose, fructose, maltose and lactose. All these sugar were considered as a reducing sugars. All those are having free aldehyde and the ketone. Non-reducing sugars, those sugars which are not having the aldehyde and ketose, free sugars at the free end. So the aldehyde and the ketose groups are not in the free end. So that is why we are calling them as a non-reducing sugars. The best example will be the sucrose. Very important. So this is also an important question. Just look at this picture. This is what a reducing sugar and this is non-reducing sugar. So in the non-reducing sugar, just identify what will be the linkage here. See here, there is a C. This is C in the carbon, there will be H. So beta. Why beta? So what is the difference between alpha and beta? What we have discussed here, alpha and the beta. So if it is an alpha, then the OH group in the downside. If it is beta, then the OH group will be in the upper side. Just look at this picture. The OH group is in the upper side. So this is why we are saying beta D galactopyranosal 1,4 linkage. So this is one sugar and this is also another sugar. Both are getting attached. What will be the linkage which is attaching these two sugars? They are having a 1,4 linkage, because first carbon of one sugar, it is linked with the fourth carbon of the another sugar. So that is why the linkage 1 to 4. Isn't it? So this is what the lactose sugar. So the lactose sugar on hydrolysis, they are giving a glucose and the galactose. So this is what a galactopyranosal and here it will be glucopyranose. So this is the linkage and this sugar is called as a reducing sugar. Those are having the aldehyde and the ketones in the free group. Free aldehyde and the ketone group. Now here it is non-reducing sugar. So this is the structure of sucrose. Just to see here the CH2 and here it is getting, both are getting related, both are getting linked, and what will be the linkage? Alpha 1 and 2 beta. Alpha 1 and 2 beta. So this will be the name of the linkage and this is considered as a non-reducing sugar. Now coming to the another disaccharide, that is maltose. So this will be the structure of the maltose. So always the question can be asked, what will be the linkage of the sugar? What will be the linkage of this particular sugar? So the maltose means, maltose is a disaccharide. On hydrolysis they gives rise to two different sugar, and these two different sugars is nothing but 2-glucose, and these 2-glucose is getting connected with which linkage? And that will be alpha 1,4 linkage. The alpha carbon, the alpha first carbon of one sugar and it will be linked with the fourth carbon of the another sugar. So that is why the linkage alpha 1 to 4 linkage. And see here, glucose plus glucose, we are calling them as a maltose, and alpha 1,4 glycosidic bond. If two sugars are attached with the which type of bond? That will be glycosidic bond. And this sugar is called as a reducing sugar. Why? Because we are having a reducing end. Where is the reducing end here? COH, which means CHO, and see here also COH, that is what? Aldehyde, free aldehyde group. Isn't it? Yes, lactose. And just look at here. And this is also a reducing sugar. Reducing sugar, another thing, that will be the free aldehyde group will be present here. So that is why we are saying this particular sugar is a reducing sugar. And what will be the linkage? Beta and first carbon, and here it will be the fourth carbon, then it will be beta 1,4 glycosidic bond will be the linkage present between the glucose and galactose Together we are calling them as a lactose. Now, and this is called as a milk sugar. The sugar which is present in the milk is called as a lactose. And now sucrose. Sucrose is a very important structure. So frequently the question will be asked from here, because there will be no free end of functional group. Either it will be the aldehyde or ketones. There will be no free end in the glucose and fructose. In the glucose, aldehyde will be the functional group, and in the fructose, ketone will be the functional group. Here there will be no free ends of the functional group. That is why we are saying the sucrose as a non-reducing sugar, and the sucrose is called as a table sugar, and the glucose is attached with the fructose, with which linkage, and here the first carbon of in the one sugar, that is in the glucose, is linked with the second carbon of the fructose sugar. So that is why the bond alpha 1 to beta bond. So this is very important, and always two sugars is linked with which bond? Glycosidic bond. So this is also very important. Now, what is this glycosidic bond? So formed between anomeric carbon of one sugar and OH of the another. Anomeric carbon in the sense first carbon. So here this between the anomeric carbon of one sugar and the OH group of the another sugar. So by this this interaction is called as a glycosidic bond. And what will be the important reaction happening here? That will be the condensation reaction. And with the help of this condensation reaction, in the sense there will be the loss of water. The water has been lost in the formation of glycosidic bond. So the glycosidic bond will be it is formed between anomeric carbon of one carbon and with the OH group of the another carbon, and whenever these two has been linked, the water group has been released, so that this reaction is called as a condensation reaction. And the examples of this glycosidic bond, so in the starch and the glycogen, it will be alpha 1,4 glycosidic bond. In the cellulose, it will be beta 1,4 linkage. So this is very important. The question can be asked from here. Now, the polysaccharides. See here, storage polysaccharides. So, in storage polysaccharides, in the storage, excess sugar has been converted to glycogen in case of human. In case of plants, the excess glucose is converted into starch. Just see here, starch which is present in the plants, they are having two different components. One is amylose and amylopectin. Amylose is in the linear structure and the amylopectin will be in the branched structure. And what will be the linkage present in the amylose? That will be alpha 1,4 linkage, and in the branched amylopectin we are having alpha 1,4 linkage and alpha 1,6 linkage. And if any compound, if it is present, to test whether there will be the starch is present here or not, that test will be called as the iodine test. Whenever the test has been positive, then it turns to blue color. So this is very important. Now comes to the polysaccharides, that is glycogen in case of animals. So the glycogen, it is highly branched structure, and what will be the linkage of this glycogen? That will be alpha 1,4 backbone and alpha 1,4 branches. So the excess glucose will be converted into glycogen and it will be stored in the liver and muscle. This is what a structure of a glycogen, and that will be, so here one sugar and here in see group of glucose is nothing but a glycogen. So see here there is a branched structure. So this is why we are saying glycogen will be in the branched structure. Now the structural polysaccharides. The structural polysaccharides, the best example will be the cellulose. Cellulose, beta D glucose unit. So all the many glucose we are calling them as a cellulose, and one glucose is getting linked with the another glucose, that will be in the means of beta D glucose units. And the linkage will be 1,4 linkage, 1,4 glycosidic bond, and this cellulose is insoluble in nature. It is not soluble. It is very rigid. It is toughest structure and it cannot be digested in humans. This is very important because of the absence of cellulase. Cellulase is the enzyme going to digest the cellulose. As cellulase is not present in the human, we can't digest the cellulose. Now chitin. So the structural polysaccharides, all these were the cell wall. All these sugars are make up the cell wall. In case of cellulose, plant cell wall is made up of cellulose, and in case of chitin, the fungal cell wall is made up of chitin. And this fungal chitin is together we are calling them as the N-acetyl glucose amine. And this chitin is present in the fungal cell wall, and also the arthropod exoskeleton is also made up of chitin. Now heteropolysaccharides. Heteropolysaccharides, so all these are, now we have discussed all the sugars we have discussed as of now. All these were the homopolysaccharides. Homopolysaccharides means what? Many glucose, many fructose will together make up the polysaccharides, many glucose will make up the polysaccharides. But here in case of heteropolysaccharides, they are composed of different monosaccharides. And what will be the best example of this heteropolysaccharides? That will be hyaluronic acid, heparin, chondroitin sulfate. So all these were the sugars just coming under the heteropolysaccharides. So lubrication, what will be the function of this heteropolysaccharides? For lubrication, anticoagulatant and connective tissue. So these are all the best example of this heteropolysaccharides. Just look at this picture. And in the sugars, we are having sugars, sugar acids, sugar alcohols and deoxy sugars. Very, very important. Just look at this picture. We are having CHO and CHOH and CH2OH. So this is only one sugar. One sugar, we are calling them as the monosaccharides. If it is a sugar acid, we are having COOH. So this is carboxylic acid. If acid is present in the sugar, we are calling them as the sugar acid. So this is the basic formula. And just look at this picture, and CH2OH, CH2OH. And if the alcohol group is present here, then it is called as the sugar alcohols. And if the deoxy sugar, what is mean by deoxy sugar? See, COOH. And here CH3, one minute. COOH, CH3 and OH, and here CH2OH. See, there will be no methyl group added in any of these sugars. But here, why we are not having the OH group here? Okay, if there will be the presence of oxygen is present in this carbon, there will be no point of having here, there will be no oxygen is present in here. So that is why we are saying deoxy sugar acids. So this is very important. So all these were the basic formula for the sugar, sugar acid, sugar alcohols and deoxy sugars. So at the second carbon, so this is very important. At the second carbon, there will be no oxygen. See, the OH group is replaced by the H. So that is what we are calling them as the deoxy sugars. Just look at this picture. We are having two nucleic acids, that is deoxyribonucleic acid, DNA, and ribonucleic acid. So this is what ribose sugar and deoxyribose sugar. In the second carbon, there will be there will be absence of this oxygen. So that is why we are saying this sugar as a deoxy sugar. And here there will be, in the sugar we are having the oxygen. As the oxygen is present, we are calling them as the ribose sugar. So that is why we are calling this as a ribose sugar. And also the amino sugar is also present. So what is meant by amino sugar? If the OH group has been replaced by NH2, here usually there will be OH group. But the OH group has been replaced by the amine group. So that is why we are calling this particular sugar as an amino sugars. Example, glucosamine, N-acetyl glucosamine, N-acetyl galactosamine, or the derivative form of this amino sugars. Now the sugar acids. Sugar acids, just look at this picture. These are all the oxidation products, which means aldehyde on oxidation, it will be converted into acid. Aldehyde On oxidation, it will be converting to the acid. So that is why on the oxidation, we can get the sugar acid. The best example will be the gluconic acid and glucouronic acid. So this is what gluconic acid and glucouronic acid. Just look at this picture, look at this formula. Here in this aldose sugar, the one aldehyde will be oxidized, so that it gives COOH, and the rest of the formula are same. So if only one side there will be oxidation taking place, then that sugar is called as an aldonic acid. So if this CH2OH, if it will be oxidized and if it is converted into carboxylic acid, then we can call them as the glucouronic acid. Okay, now if both the sides are getting oxidized, then we are calling that particular acid as the aldaric acid. So all these are the very important sugar acids. Now, what will be why do we have this carbohydrates? What will be the biological role of this carbohydrates? Primary energy source of this energy source will be the glucose, which is nothing but a carbohydrate. And the structural role, to maintain the structure of this plant and the fungi, this carbohydrates is very important. And to store the excess glucose, carbohydrates is very important. Components of this nucleic acid. In the nucleic acids, we are having a ribose sugar. So for the component of the nucleic acid, carbohydrate is essential. And cell recognition. One cell has to be react for the another cell's signal. It has to receive the another signal, and it have to respond for that signal. So that is what a cell recognition. For the glycoprotein and glycolipids are there. The protein associated with the carbohydrate is glycoprotein, and the lipid associated with the carbohydrate is glycolipids. For all these processes, carbohydrates are very important. So as of now, we are completed majorly the most important topics, and you have to, what will be your work will be, so you have to what are all the books you are having, you have to refer once again the carbohydrates, and you have to strong with this particular chapter. Now, which one of the following carbohydrates will give a negative Benedict's test but positive Wallis test? So, when it comes to the exam point of view, all these tests are very important. So, your homework will be analyze what is Benedict's test and what is Wallis test. Right? So you have to test whether you have to identify what is, you have to study about these two tests and you have to answer. So the answer will be sucrose. Wallis test is general test for the carbohydrates. So the general test for carbohydrates is Wallis test. And the Benedict's test, as we have studied in the previous slide, to check whether it is reducing or non-reducing, that will be the Benedict's test. So the Benedict test, if you know what this test is for, you can easily answer those questions, because sucrose is non-reducing. So obviously Benedict test will be negative and Wallis test will be positive. Why? Because it has the general test for the carbohydrate, whether it is a sugar or not. So the answer will be sucrose. The number of optical isomers possible for aldohexose. So to identify the optical isomer, you have to know whether how many chiral carbon will be present. So how many chiral carbon will be present in the aldohexose, then you should know what will be the n, and by replacing the n you can easily answer this question, and the best example will be 16. And the answer will be 16. So in the aldohexose, usually there will be 4 chiral carbons. Now, glucose and galactose differ from each other in the configuration. For this, you should know what will be the structure of this glucose and what will be the structure of galactose. And you, by comparing these two structures, you have to identify at which carbon there will be the difference, and that will be the C4 configuration, because there will glucose and mannoctose will be the C2 epimer, and the glucose and galactose will be the C4 epimer. This is very important. Now, which statement is correct regarding the fructose? So, it is an aldose sugar. No, it is wrong. It shows an optical activity. It is a reducing sugar. It does not form a cyclic structure. Again, it can form the cyclic structure. It is a reducing sugar. Of course, it is reducing sugar. It does not show optical activity. No, it is show the optical optical activity. So, the best answer for this question is, correct answer is, it is a reducing sugar. Now, the glycosidic linkage present in the glucose. So, the glycosidic linkage of most of the sugars are very important. This can be easily expected in the exam. So, that will be beta 1,4 linkage. So, next question will be, which of the following pairs represent anomer? Represent anomer, which is considered as an anomer. So, there will be glucose and fructose. Glucose and galactose are an epimer. Alpha D glucose and beta D glucose. Ribose and deoxyribose, and which pair is considered as an anomers? That will be the alpha glucose and beta glucose. Now, which carbohydrate is stored in liver and muscle and has the highest degree of branching? That will be none other than the glycogen. So, and just know what will be the linkage of all those. Now, which one of the following is a heteropolysaccharide? That will be the, so all other will be the structural polysaccharide, that is homopolysaccharide. One will be the heteropolysaccharide, that will be the hyaluronic acid. Now, the sugar present in DNA differs from ribose by absence of oxygen at which group? Absence of hydroxyl group at the C2. There will be no oxygen. The absence of this hydroxyl group is being replaced by the hydrogen group. Okay, that's why the deoxy, the name. Which statement about sucrose is incorrect? It is a non-reducing sugar. If it is a right statement, it contains glucose and fructose. It is a right statement. It shows mutar-rotation. Both anomeric carbons are involved in linkage. And what will be the incorrect statement? It shows the mutar-rotation. So, what is meant by mutar-rotation? The sugar which undergoes a mutar-rotation, and those sugar that definitely have free anomeric carbon. Those sugars which are having the free anomeric carbon, all those sugar will be undergoing the mutar-rotation. As this sucrose is not having a free anomeric carbon, there will be no, it doesn't undergoing a mutar-rotation. Okay, so in today's class we have completed the carbohydrates, and it is not sufficient. You have to study more about this. Thank you so much. We'll meet you in the next class.